Today, I will present my study titled Semaglutide-Induced Neurodisruption via the Gut-Brain Axis and Restorative Effects of L-theanine and Hesperidin. This project investigates how the obesity drug semaglutide may influence the brain through the gut-brain axis and how natural compounds might reverse those effects. Semaglutide, also known as Wegovy, is a GLP-1 receptor agonist widely prescribed for type 2 diabetes and obesity. It mimics GLP-1 to enhance insulin secretion, suppress appetite, and induce sustained weight loss. However, recent clinical reports now describe mood changes, insomnia, and cognitive impairment among patients. Because GLP-1 receptors are also found in the brain, this suggests a possible mechanistic link between the gut and the central nervous system. Given its profound benefits, including glycemic control, obesity management, and reduced cardiovascular mortality, semaglutide's use is expanding rapidly. However, this is paired with a growing concern for its adverse effects. Beyond the common gastrointestinal issues like nausea and vomiting, patients face serious risks, including a significantly higher risk of intestinal obstruction and pancreatitis. Despite these known serious physical risks and the emerging reports of neurological side effects, there is a major research gap. There is limited mechanistic evidence for how semaglutide affects brain function through gut-brain signaling pathways. This led us to our hypothesis that semaglutide may indirectly suppress neuronal activity through intestinal signaling, and that such effects could be reversed using L-theanine and hesperidin, which are two natural non-invasive compounds. To test this, we developed a transwell co-culture model of human intestinal cells, which is called CACO2, as well as neuronal cells called SHSY5Y. Semaglutide at 50 nanomolar was applied only to the intestinal cells for 48 hours, where indicated L-theanine and hesperidin at 100 nanomolar each were co-administered. After treatment, we analyzed changes in the neuronal cells using RNA-seq, qPCR, and ELISA. In neurons indirectly exposed to semaglutide, we observed a significant decrease of GABAergic genes and circadian genes. These genes are essential for inhibitory neurotransmission and sleep regulation. This molecular suppression provides a direct biological explanation for the clinical symptoms of mood disturbances and insomnia reported in patients. Next, we examined whether natural compounds could reverse these changes. L-theanine is a green tea-derived amino acid that enhances GABA receptor signaling and promotes neuronal relaxation. Hesperidin is a citrus flavonoid with antioxidant properties and circadian regulatory effects on neuronal function. Both L-theanine and hesperidin individually restore part of the gene expression, but the combination treatment produced the strongest recovery, returning GAD1 protein levels almost to normal. This indicates a powerful synergistic effect, enhancing GABA signaling and regulating circadian balance simultaneously. In summary, this study provides the first experimental evidence that semaglutide can indirectly suppress neuronal gene expression via the gut-brain axis. Moreover, co-treatment with L-theanine and hesperidin can counteract these adverse effects. This suggests a promising natural therapeutic strategy to mitigate central nervous system side effects in GLP-1 receptor agonist users. So why does this matter and how can it make a difference? First, it transforms a clinical observation into an understood biological mechanism, moving us from simply seeing a problem to understanding its root cause. Second, and most importantly, it proposes a practical and accessible solution. We identified a potential non-invasive therapy using safe, natural compounds that could significantly improve the quality of life for millions of patients. This work opens the door to developing adjunct therapies, ensuring people can benefit from the life-saving effects of semaglutide without suffering from its debilitating neurological side effects. Of course, this work also has its limitations. Our in vitro model lacks the full physiological complexity of the human gut-brain axis, such as microbiota, immune, and endocrine interactions. Future studies must include in vivo validation, behavioral analysis, and investigation of microbial or inflammatory pathways. Through these next steps, we hope to advance safer obesity treatments and reduce the neurological side effects of GLP-1 therapies. Thank you.